from the audience?
So this is directed, I guess, primarily at Phil. I'm interested that you have sort of standard RNA-seq scripts and these kinds of things that you publish, especially when you're a national scale infrastructure. And I imagine you have all kinds of different people working on different organisms and different whatever. I work in a much smaller center, and I still find that even on something, standard like RNA-seq, that, one workflow is never going to work for everybody. There's always going to be, this genome's got a GFF in a funny format, or this one's too big and SAM tools doesn't work, or it's, two species plant pathogen together, and so you have to process it differently. How do you actually support that many clients without having like a million parallel versions of your workflows?
Sure. So firstly, there's a few caveats that come with our bioinformatics analysis. The buzzword I usually try to give it is like a descriptive bioinformatics analysis. So we don't do differential expression analysis. We don't do any really project-specific analysis. We take, for RNA as an example, we take it up to alignment and gene counts. Secondly, we have a list of supported organisms. It's great that you mentioned Illumina igenomes. We use exactly the same resource. It's fantastic. And so we simply say, if your organism is in the igenomes reference that we have already, we'll run some analysis for you. If it's not, sorry, you're on your own. One of the great things about building this pipeline, one of the big focus for us was to make it easy for groups to run it themselves. So in those cases, we say, look, here's the pipeline we would run. Here's the documentation telling you how to run it. You plug in your own reference genome and it will work. And yeah, I mean, that's pretty much it. Most people seem pretty happy with that approach.
Thanks.
I have a question for Phil. Being able to look at all the samples, did you find out the problem Illumina had with the cross-contamination within the lines?
With the what, sorry?
Do you know there is I think it's with the HiSeq 3000, there is a problem with the tags which produce cross-contamination within the samples of the same line?
You mean the pattern flow cell hopping?
Yeah,
problem?
yeah. Were you able to see that?
Yeah, sure. So we don't have the HiSeqs which are affected. The HiSeqs we're running is not patterned flow cells, so they're fine. But we do run the X10, which is patterned, and we've just got the NovaSeq, which will also be affected by that. I mean, I'm sure everyone in this room is probably aware of the situation. I know Stephen here has written an article about it on QC fail. We have seen it. We have gone back through all the projects when it became a big problem. People started asking us about it. We went through all the projects which could be affected by it and checked. Thankfully, most of the sequencing we did on our pattern flow cells was X, and usually that was whole genome stuff which we weren't multiplexing very much. So we got off fairly lightly. With the NovaSeq, that won't be the case, but we're going to try and do unique dual indexing so that we can remove those contaminants straight away at the multiplexing stage.
We also saw it in a few projects in RNA-seq where the users introduced a specific mutation, and then we were running it on the 4000, and then we saw this cross-contamination, but it affected with RNA-seq with this quantitative method, and then it affected maybe 0.1% of the reads. So I think it was easy to neglect. But if it's variant calling and you're hunting for very rare variants, I mean, then it's really a critical issue. But otherwise, I think it's not so critical. But it's happening for sure.
Also, just to mention on that topic, Tim Booth at Edinburgh gave a really nice talk at BOSC, a lightning talk yesterday, where he's developed a tool which runs on BCL files and can run while it's still being generated off the sequencer, and that does a sub-sampling approach just to flag any flow cells where that may be a large issue. So it might be something that's interesting. I can't remember the name, but I have it written down somewhere.
Manju Kelly from the EBI. I've got a question for Lennart, really, and the CCDB. You say you're storing all of the pipelines and the results from the analysis sort of forever. Do you have any external users or collaborators? And if so, are they okay with you storing this stuff?
Yeah, actually, for the storage, we don't make a difference between. We have externals. We have even companies who are using us as a service. we don't make a difference. Just, I mean, we have core lab, we are getting money for it, so you just have to pay more for it. And yeah, storage is more expensive and all the services. But we're also keeping it for them. But of course, we have also a lot of users which just need only the data, and then they have 2 months or 3 months of time where they have to download everything. And for them, we are doing also some basic QC steps, of course, where we also track them, and the QC reports we are also keeping forever, but not the actual data to check what's going on. But yeah, our actual user base is even larger, that we have also some externals from whole Europe. Sometimes we are getting requests. But we are more expensive, we are Switzerland, so.
Just to echo what you said, really, we work with industrial partners, pharmaceutical companies, and we have a policy of backing up all the data for 6 months, I think, maybe, or maybe less than that. So users basically sign up to that when they come to us, and we take security and privacy pretty seriously, and so they usually go through what exactly we're doing in quite a lot of detail. It's something we're always evaluating though, and one of the things we're thinking about is changing an infrastructure in the future, and potentially using cloud infrastructure in a few years, and that will be something we'll have to come back to again for sure.
Maybe it's interesting in this discussion, what's your definition on of reproducibility? I mean, what I stressed at the end, I mean, should you get exactly the same results in 10 years, or is it not necessary? Is there someone who wants to comment on that? Maybe.
Yeah, I don't know about reproducing it to 10 years, so maybe it's better to resequence and repeat the total experiment, because we are further in the technical development. But what we have sometimes are projects which are stretching over years. So the data is collecting over 2 years with different aspects, and they do different assays on the same conditions. And I mean, for us, it's always like, okay, you should process it if you have RNA-seqs from 10 different perturbations, that you actually can combine them and also run them and compare them still after you have collected the data for a year or two, even if your R versions are changing in between. And that's a little bit of a struggle where we don't have an end solution for, because we want to keep it and not every time we have a new data set reanalyze everything.
Sure. So we use a similar, I was pleased you mentioned the module system. That's currently our primary way of handling this kind of software versions and everything. And we deal with it by doing these versioned releases of pipelines. So when we deliver a project, that pipeline is tagged at that version that's on GitHub, and so we can rerun specifically that version of the pipeline. It will load the same software versions through the module system. What we're still not, like as you said, it's not perfect, that situation. So we'd like to use a container system. Like most people, we can't run Docker on our HPC system. However, as of a few weeks ago, we finally got our sysadmins to install Singularity, which is a container system pretty similar to Docker, but which most HPC cores can run. And that's something we're now actively working on. And the hope with that is that we can run the analysis within a container and deliver that minimal container alongside the data. And then in 10 years' time, hopefully, as long as Singularity support is still around, people will be able to get back in and rerun exactly the same thing. I just want to give another kind of tip on Nextflow. One of the main reasons we use that is it's compatible with all of these different systems, and we don't touch the pipeline itself. We just have different config files which are wrapped. So it runs both. We just specify a different flag, and that switches from using a module system to Docker or Singularity or whatever else, which is a really nice feature. So we're hoping we're future-proofing ourselves and not having to reinvent the wheel in the future for that.
Quick question for Phil. You mentioned that you just received the ISO certification for your bioinformatics analysis. Could you tell us briefly what the requirements are to get that kind of certification?
Sure. So the ISO certification isn't specifically for the bioinformatics, it's for the entire facility. And we received it quite a few years ago now. So it's quite a flexible certification, which is nice. It means we can add things into it without too much paperwork. And so the bioinformatics was done like that. It was a natural extension, and it's basically an optional extension for us. In order to get the validation, we have to show that everything can be audited, so that we never throw a log file away, basically. It doesn't necessarily have to be easy to access, but as long as we can show it's there if necessary. And we also have to demonstrate that we do these validation checks, so every time we make a change, we are checking that it doesn't affect the results too much, and if there are effects, we document them. And also we have to have a complete documentation system around the system. So we used to use Google Docs, and we now use AM Systems, which is a documentation system. And there's like an operation procedure for running every analysis pipeline, which takes you from start to end, and those documents are also versioned. So it's lots of documentation.
I'm curious to know how the QC reports are used and who gets to see them. Are there hard metrics in place for, say, passing and failing? Or is it always a visual inspection? And do you pass the reports on to scientists, regardless of whether things have gone really well or whether there's been an issue?
Yeah, so we have hard metrics, hard levels, for instance, kind of alignment rates, duplication rates, etc. But they are always optional. So they can be a warn level where we'll tell the user, this looks a bit dodgy, but it's probably fine. Make sure you have a look. We have failures, and they run on different levels. So we have a guarantee for the users that if the samples passed QC reception control when they came into the lab, and then they fail our bioinformatics QC, that will rerun the library prep and sequencing. And we try that once. If they're still failing, then we usually tell the user, but we usually end up delivering that data anyway. The reports always go to the user, and we also store those reports internally as well. And there's also a visual check as well on top of those automated checks, because they're never sufficient.
So I have a related question for both speakers. From the point of view of users, how do you believe that this kind of work is perceived? So you both described infrastructure that took a long time to set up and develop, so it's a big investment. Do you have the feeling that your users understand the value of all the work that goes into this, and they appreciate the results, appreciate the effort, or do they just see the benefits without realizing everything that goes towards that?
Most of our users are predominantly based in the lab and have limited exposure to bioinformatics. So I think they do appreciate the laboratory side of things, and I think probably, hopefully not the majority, but certainly a large proportion have either no idea or don't care about the bioinformatics analysis. One of the main problems we had, which led to us rewriting our recent pipelines, is that lots of people basically didn't trust our analysis or couldn't replicate our analysis, and so would routinely discard what we produced and rerun it themselves anyway, which is just a massive waste. So that's why we were really keen to build a pipeline that other people could run themselves. Whether it's worth it, I think it's worth it even if no users use our QC metrics, because we can use them internally, and we can spot internal errors. And I think it's a good thing. Hopefully, it's a good thing for kind of science as a whole, even if the users don't appreciate it.
Yeah, I mean, in our case, it's mixed. I mean, we have also to deal with a lot of medical doctors, and for them it's even harder, I think, to see the point and and to appreciate that we developed our own tools to improve all this documentation and reproducibility options. But otherwise, I mean, we are also using Sushi, for example, for courses to make students aware that we have our own solutions to create a future user base and so on. They fully understand, and for example, some people are also working then with Galaxy and so on, and then they really see also the difference, how easy it is in to use our customized tools in comparison. I even met a guy here at the conference who was originally a biologist, but now was hired as a bioinformatician in a company in Munich, and he is really missing Sushi because they have no workflow manager solution or so on. So there are a lot of people who appreciate it, and they see the point that it took a lot of time and so on to develop all this stuff. So it's a mixed situation. But it's still encouraging, I think.
In that vein, working with the users, you have automated and control all of the bioinformatics, compute-heavy parts, but then data gets loaded into Excel, cells are marked, and things are typed into Word, right? So that's actually the interesting part. I'm sorry if I'm offensive here, and there's very little things that you can do. And my own, actually, I had people walking out of my lecture on reproducibility when I said, now let's look at the code. So how do you do that? I mean, do you have some handle on that? Can you support people there? We're talking about technology here, so shut me out if I'm going too far off topic, but it'd be interesting to see your point.
I definitely sympathize with that point. We've had a lot of requests to incorporate automated differential expression into our analysis, and I just keep saying no. The idea of some of the horrible things that people can do with the data terrifies me a bit. But there's limited amounts you can do. I mean, obviously, once we deliver the data, that's it. It's gone. It's out of our hands, and the group has to take responsibility for that. The best we can do is encourage people to follow good practices, we provide some training ourselves, and there's also lots of training within SciLife Lab, mainly by the bioinformatics facility. And the other thing that's nice about being embedded in the sequencing facility is I think we probably have the opportunity to have contact with the groups at an earlier stage than perhaps many other bioinformatics courses, because we have project coordinators who have meetings with every single user when they come in with a new project, and we often get invited to those meetings, and I can sit there and say, no, you need more replicates.
Yeah, but maybe instead of using Excel and so on to filter results on their own, that's the purpose why we also developed some interactive tools then with Shiny that can prevent it a bit, and then it's a bit more controlled than their Excel acrobatics, what they are doing then afterwards. So this is our solution, or how we try to educate the users to use this.
Can I come in with a comment? I would say that training on experimental design and coaching people to involve you in experimental design is one of the most valuable things you guys can do. And not only will it help your users, but I think it will help you as well, because then you won't get badly designed experiments that you then have to deal with and cringe when you give the results back.
Yeah, it's a main advantage if you're working in a larger core lab, which includes the sequencing and the bioinformatics. Then it's automatically a kick-off meeting is always together, where you can prevent that people are using whatever, only duplicates or whatever, not even any replicates and so on, to optimize a bit the design and to prevent that they deal with too many confounders in the same experiment. That's the main advantage. Also, that we have the wet lab people next door. So also for all the QC steps, we are not only reporting, we can also actively participate in improving some stuff if there are certain biases in the data and so on. I think sometimes bioinformatic core labs are a bit isolated from the wet lab part, so that it's often, I think it's a big advantage to have everything together. And in our case, everything on the same floor.
I actually have a question for you. I've kind of had the idea of Shiny in the corner of my eye for a long time, and we've talked about setting up Shiny apps for our users, but haven't actually done anything with that yet. And I really liked the thing you touched on, saying how you can export a static R script which would replicate that interactive analysis. Before working in Sweden, I worked with Stephen in the Babram core, which is a much smaller core, and there's Simon Andrews, who I think has been involved in this group a lot before. He's developed a graphical tool called SeqMonk with the express intention to give users the power to run their own analysis. And it's fantastic, but it's also very difficult to replicate. because it's a graphical tool and there's lots of pointing and clicking, and there's only so much auditing you can do. So the idea of an interactive tool where you can also export a reproducible script, I think is a fantastic idea. And I wondered if you could elaborate on what those apps are and maybe how much they're used, if you know.
Yeah, so if we run differential expression, we are creating something which is called then a static report. So it's an HTML report which includes a lot of plots and which are not interactive, of course, and also the magic table, which is the Excel sheet with the differential express streams. But in addition, when we create this report in the Sushi app, it creates also an R data object, and which is used then for something which we call then the live report, and then this is the Shiny app. In the Shiny app, we can reproduce all the plots which are in this static report. But for example, if there's a volcano plot, and people can then click on a certain dot and can check what kind of gene it is. You can create for every gene box plots across your conditions. You can create a lot of different kind of heat maps and so on. You can run sample clustering with kicking out 2 outlier samples or whatever. So all these things are possible with this live reports, which are kind of complementary to the static ones. That's our idea right now. Because we are also a bit, I'm also a bit tired of this user requests who want to have for a specific gene set for their data a heat map. I want to educate them how to make their own heat map in a very easy way, and or Venn diagrams or whatever. All these things which are always repetitive, which always come back, and where many users are asking for. All the things are, I think, Shiny is a very powerful tool to get rid of this work and to educate them to do that, and then use these apps. So that's the idea: static report, live report, how we call it. Is it released? No, it's not published. No, it's only for internal usage. It fits to the structure, to the data structure we are using in our R package. That's why I'm not sure if we will ever. Yeah, we have it also under version control. It's also in subversion, I think, not GitHub, but it would be available. But we could make it available if it's useful. I could also make a live demo. Later, maybe. More questions?
Maybe also, I remembered you asked over there about whether we always deliver reports to users, and it's true that we deliver the multi-QC report, which is the one I kind of demoed, but I kind of felt guilty after I replied, because we have lots of other information which we don't deliver to users as well. So in the genomic status website we run, which collects everything from limbs, then we have like caliper images, we have lots and lots of lab-based information, and we are slightly more selective about which bits get delivered to the user there. But usually if users request it, which basically never happens, then we're happy to hand over that data.
So I think a natural evolution of the discussion is it's increasingly a requirement when you publish, because some of us don't have the luxury of say that we're not interested in doing the analysis. We're asked to go all the way to final analysis and then helping with publication and dissemination of the data, which is increasingly a requirement, right? We're being asked, authors are asked to deposit their results to publicly available repositories. And the natural evolution of this is that it may also become a requirement to publish the quality control reports, right? Not just publish the data, but also publish some kind of information that shows that the data was generated in a way that makes sense, and the input materials were good, and the analysis process worked, and so on. So do you think that, first of all, it's a good thing that we're going in this direction, and there will be standards, or what is the mechanism for doing this, for publishing not just results, but also supporting information about the results generated by some of the pipelines that you've described?
Yeah, I think it's definitely a good thing. I think it has to be the way things go. On Twitter, the other day there was a Twitter poll, I can't remember who launched it, asking how many people had been infected by not knowing what reference genome was used for an analysis. and that was very commonly a case by by the responses. If information as obvious as that isn't included, then that's clearly a big problem. And I don't think these data files are very small, they're very easy to handle, so I don't think there's need to be any limit on that reproducibility reporting. And there's been a few papers coming out recently where there's been accompanying GitHub repositories with full source code for the entire analysis. And although it seems like a lot of work, I hope that as this becomes a more popular, talked about topic, then it's the kind of thing where if groups start off using like that approach, kind of one doesn't have to be a repository, but one kind of documented location from the start, then hopefully that will become easier and easier.
It's also a question for the audience. I mean, does anybody have experience with standards or mechanisms to disseminate, to distribute quality control information in a standardized format? Or is it too early for this?
If anyone has any ideas and thinks that multi-QC could be able to export that kind of thing, then let me know. I'd be interested.
Any other questions, comments? Okay, then I think we can thank our speakers again, all of them.